Welcome to Akhand Vidyashram, the first institute of impeccable wisdom. Let's discuss about DNA in the plant kingdom and compare it with animal kingdom and bring out something which is difference between plant DNA and animal DNA. So plant genetic structure, let's start with. Let's start for Kapil Vriksh, the tree which has roots in heaven, and things have come because everything what we have today on earth or in the universe is from divine kingdom. And in this particular picture you can see very well that whole evolution of chemistry started with three atoms: hydrogen, carbon, oxygen, and they formed methane plus oxygen, then water and carbon dioxide. And these three things, along with methane, came down as three molecules, and then the process started for the production of plant kingdom, mineral kingdom to plant kingdom. Now, in plant kingdom, we get before, as I told you in the beginning, it was methane and carbon dioxide which came down with the cloud down. And at this juncture, let me make it very clear. To start with, there was 97% carbon dioxide on Earth also, along with Mercury, Venus, and Mars. All the four inner planets between Sun and asteroid belt had 97% carbon dioxide, and along with water, it came down. But this is the plant kingdom. We started as unicellular organism or asexual reproduction to sexual reproduction to the one which converted in the presence of sunlight, carbon dioxide and water into glucose plus oxygen. As a result, 97% carbon dioxide was converted into a 21% oxygen, essential for biological life of living cells or human beings or animals on the earth. And glucose was also required. But meantime, carbon along with hydrogen was converted into nitrogen, and that led to the formation of glucose fatty acids. And with nitrogen coming to play a role, it was formed to produce amino acids and protein to the second block. And not by talking about the third block. But before that, let me clear, in animal cells, glucose plus oxygen was consumed to produce carbon dioxide and water. As a result, that equilibrium between carbon dioxide and water which came from up is also replaced so that we have uniformity around the earth in the form of maintaining this. The atmosphere takes care of it, and the production of carbon dioxide and water from animal cells and plant to glucose, and this its cycle is the most beautiful thing which I can see is full of divine mathematics, including Divyank. Now come to the question of glucose was the one which was used in mitochondria for cell respiration to put carbon dioxide. Second, it is also stored as starch in seeds for reproduction of sexual reproduction in plant kingdom. And third thing, it was also converted into lipids or fatty acids, amino acids, and products thereof thereafter. So this is again Trinity, which I'm talking about. One getting into three is a primary product of universal consciousness. Now, the synthesis, as I told you, carbon plus hydrogen became nitrogen in volume concentration, which can be called 1600 degree. In that temperature, it was formed for the next production to convert 97% carbon dioxide into 78% nitrogen, which is unreactive. It's a molecular form. Along with oxygen, now we have atmosphere like that. Now there comes the question of fifth element which comes to play a very important role. As I've written in this, nitrogen was formed from carbon plus hydrogen, and I do believe phosphorus may be formed from silica plus hydrogen. As you can very really see in this particular thing, nitrogen is 7 and phosphorus is 15, 7 plus 8. And then put together becomes the next one to go on. So I do believe silica 14 plus 1 is 15, and that could be the reason which science can try to figure it out whether phosphorus is found from silica and hydrogen. Now come to the question of phosphate molecule. You have phosphorus has got 15 protons and oxygen 32 protons. Now I'm bringing the concept of proton in new game to understand divine mathematics very clearly. If you take phosphate as such, you have number of proton only 47. But if you take monophosphate, which is used as adenosine monophosphate, as that you have 49. You can see the picture very clearly. Is it trivalent? Carbon can get 4. 4 oxygen can also to produce, or 4 hydrogen to produce methane. Now the comes to the deoxyribose. It has got 5 carbons, 10 hydrogen and 4 oxygen, and number of proton is 72. Now we are moving to the energy molecules. We have come from ATP, ADP, AMP to this stage. I'm just repeating it, from glucose to fatty acids to amino acids to nucleic acids. Energy molecules ATP, ADP, AMP. Now we are going to the third DNA stage. So how it further goes, with this background only we can understand the concept which I'm going to talk about it in DNA, which is unique, has not been done by anybody else. I'm not just trying to copy what others talk. Let's give something food for the thought so that our knowledge can go on further. So number of protons in deoxy is 72, as you can very clearly see. I have shown that in ADP also. Now adenosine is got 70 protons, as you can see very clearly, 5 carbon, 5 hydrogen, 5 neutrons. That is the formula. Now going to come to know 4 nucleic acids. Number 1 is the thymine. It's a chemical formula is carbon 5, hydrogen 6, nitrogen 2, oxygen 2. The number of protons in this is only 66. Carbons 30, hydrogen 6, nitrogen 14, and oxygen 16, 66. Please make a note. Thymine has got 66. Adenine hat, you have already seen. Now cytosine has got carbon 5, hydrogen 5, nitrogen 5, oxygen 1, and the number of protons is 78. Fourth in the line is guanine, and you have 58 protons from 4 carbon, 5 hydrogen, 3 nucleic nitrogen, and 1 oxygen. Now remember these things because they are going to play an important role to bring forth something which has not been explored till date. Now in this background, let me, I've explained in ATP also, I'm explaining again. Deoxyribose has got 72 protons. Phosphoric acid has got 50 protons. Two side chains of phosphoric acid and glucose is 244 or 122 into 122. This is a formula which I do believe has very important role. But again, this formula has also something unique to talk about it when we go further. Now let's talk about something unique between adenosine and thymine is a one combination we are going to see in DNA. Adenosine has got 70 protons and thymine has got 66. Together they are 136 protons between these two itself. You can see very clearly, and they have a bond of 2 hydrogen bonds between these two. You go to the next one, which is a combination of cytosine and guanine, and you see very clearly the number of protons in cytosine is 78 and guanine is 58. If you add together is again 136. Upper one is also 136. This is also 130. Upper has got 2 hydrogen. This is the 3 hydrogen. This 2 hydrogen bond and 3 hydrogen bond is also according to Divyank. But isn't it interesting that number of protons in both the combination equal? Has anyone in the world ever looked at it from this angle? to the best of my knowledge, I do not see. The number of strands in the OSCO, as I told you, is 10.3, and with which I'm going to see as we go on to understand the concept of DNA little as we proceed. Now comes the question of plant DNA. Now, plant DNA and human DNA, B double helix, and this is almost identical in structure. But the number of difference between these two, I say, science says the number of strands could be 7 to 8. But as far as I'm concerned, it's a plant kingdom, which is a basic structure. It should have only 7, not 8. So I'm making one thing very clear. Perfection, according to me, should be 7 because the divine number as the manufacturing process of divine nature itself. Earth is made of 7 parts. Antriksh is made of 7 parts. Heavens are 7. Days are 7. Colors are 7. Sun has got 7 horses. Everything is 7. That is 7 is basic structure. From that I see in plant kingdom as 7. But when it comes to the human DNA, they say it could be 10 to 12. But I do know it is not 10 to 12. It is 10.34419. So now let's making it very clear. Plant DNA has got 7 strands and human DNA, perfect one, has 10.3441 strand. That's it. And the picture is there. And as far as nucleic acid combination, you can see it very clearly. Both the chains are deoxy plus phosphoric acid, and in the middle you have adenosine and thymine, a guanine and cytosine. You can see this picture very clearly, and I think that is the structure which I'm talking about. But this is a basic structure which you have to keep. Now understand the basic structure a little further. Let's go to human BXX to understand the concept much more clearly, because both are identical except for the strands. Now, this is the DNA structure of a perfect human DNA, and that is structured as diagonally cut. But let's go further. Now, as you know, human B double helix was first time got together as a structure by James Watson and Fossecric, and both of them got a Nobel Prize in 1953, which is almost 70 years back. And that's the time when DNA structure became popular and genetics started taking new shape. And I'm very happy they've done a wonderful job of designing, creating, proving the structure of human builders by both of them. But of course, there are certain controversy with respect to other people. But I do know the science as such, unfortunately, is not a one-man show. If there is only one aspect which comes to the divine mathematics or divine science, which is only single, only one person is involved without any second part taking part in any form. It is Shivbhushan Sharma, who gave the world Divyank for the first time in history of mankind, based on the information got from Vedas, Upanishads, and Indian Akhand Bharat scriptures, and built on based on modern equipments to find the last part of 0.34419. And this particular structure is a journey of so many years to come to the stage. But at this juncture, let me make it very clear. I could go to Divyank primarily because mathematics available in the world was man made, not as divine as I wanted, and I wanted looking at perfection. I did not find that perfection in algebra, geometry, and other things, what was taught to us in higher mathematics in school, college, and university. That was the first thing which I thought that is defective. Divine mathematics is different, and looking for the divine mathematics within period of 60 years, I could come to Divyank very clearly. But that is one man show. But other science, whatever is talking about, is man-made concept which has gone over and over from ancient Indian Brahminic divine mathematics to the man-made concept. And now we are following only man-made concept. We are not following a divine. As a result, we have all the deficiencies. So even the people, many people get together, they know in one form or other, we come to bring certain people into name and show it. And here, these two names stand out as far as this is concerned. Other two people names are not, but I know in this, it's a joint venture of whole humanity of science. So I think everybody is involved. When I say Divyank, I owe it to ancient Indian Brahmins on one side, which gave a divine mathematics, and modern scientists and mathematicians made me think rationally and bring it to that stage. So I have to give credit to everybody. But bringing to this stage, unfortunately or fortunately, is the one man consistent look for perfection and getting it. And that is what I want next generation to start working on science with Divyank in mind, not with the man-made concepts. That is one point which I'm making very clear. This I say. These are the picture who brought it. Wonderful. Thank you. Now let's move on. Let's now even look at this particular structure. B double helix, what they talked about it, and what I'm going to talk about it. B double helix based on Divyank to prove it that we should have that kind of precision of perfection. Let's not work on disease models. One point which I'm very clear, when we are working a disease model, peripheral work, multiverse, you'll have all the confusion. Every child is confused in the world. But give them the perfection of the perfect model, which divine designed it, and then move on to a level that research will be good, but having consistent constant of divine nature is more important than man-made vibrating, fluctuating, fleeting concepts at all. That is the point which I'm making it clear from my work. It's a one-man show to bring into flavor perfection at every level of consciousness. Now, let's talk about what they talked about it as a B double helix known to the world, in that the width is 20 angstrom, height is 34, each strand is 3.4, number of strands is 10, minor groove is 12, major groove is 22. There are two enclosing chains of deoxyribose sugar and phosphoric acid chains, opposing 3 and 5 on top and bottom are the one which is according to Divyank. Nitrogen chain adenine and thymine is a one combination. Guanine and cytosine is another combination. Binding chain of 2 and 3 hydrogen is another thing which is also according to Divyank. Now this is a picture which world knows is something which Watson and Crick has given credit to. Wonderful. Thank you very much. Now in this particular picture you can see both the sides you have phosphoric and deoxy, and center you have nucleic acid. This picture I have already shown. Let's move on to the next stage to understand things very clearly. And this is the structure of each nucleic acid. The bonds 2 and 3 is something which I have already shown. Now, this is the double bond between adenine and thymine. You can see two bonds in this picture very clearly, NH and NO. Now, within guanine and cytosine, you can see three bonds, and that is picture to 2 and 3. Now you can see very clearly how the binding of 3 hydrogen atoms is. You can see 3H here also. You have seen 2H in the other one. Now I'm going to give the design done by Shibhushan Sharma for Divyank design of B double helix. In this picture you can see as such. You can see the number of protons, neutrons, everything is very clearly. You can come back to that, but let me give you the summary in the next picture. In the picture, the difference between the known model to this model, they said 20 angstrom, I am saying it 21 angstrom. That is according to Divyank. Each strand is 22 angstrom. They don't talk about it. Number of strands is 10.34419, not 10. Height is 33.9854 angstrom, the perfect one. Minor groove of 12 angstrom is absolutely same. Major groove 22, no problem. 2 sugar phosphoric back chain, no problem. Opposing and 3,5 carbon, no issues. Central nitrile adenosine thymine group, same. Guanine and same. Binding chain of 2 hydrogen, same. Bond of thymine is 2. And binding of 3 hydrogen between this other thing. But what stands out is instead of 20 width, it's 21. Instead of 34, it's 3398. Instead of 10 strand, it is 10. And the groove of that is exactly according to them, and nobody can find fault because I'm talking about the perfect model. If we teach the perfect model, they remember only, and every child has got same thing. There will not be confusion in the classes, and that is why I'm talking about perfect models at each and every stage for entire world one by one. Now this is the structure which I'm also showing. Now other two structures which scientists have given. One dice, both are longitude that cross sections. But you can see 10 in upper one, 10 on both the sides. But same structure which I have given of Divyank, you can see the structure more or less the same. If you see the right picture on top, the width that lifts is 22 and 21. You can see in this also. Inside you can see 10 strands and I'm showing 10 strands. They also have central certain thing which has not been spoken about mathematically. I have put 5 inside, which is 0.34419. So this structure of Divyank and the structure of DNA, human Divyank, is identical. So I do believe the perfect human B double hex is based on Divyank, and that should be the thing which world should need. It is thanks to Watson and Crick they have brought that structure. Now I am taking to perfect. Now let the world talk only perfect model, so that everybody that uniformly knows throughout the world that we are not talking about disease. Let that be the medical profession to do it. Disease people, patient, doctors who are going to do it, let them do it. But as far as basic knowledge of anatomy, physiology, biochemistry, psychology is concerned, let that be based on Divyank, Divyank ratio, Divyank sequence, Divyank infinity, which I have spoken about it. I'm going to speak again in the new form of book as Divyank Sutra also, which the book is due as I'm proceeding with this thing to making that another thing, which will be like one lot for the entire world, and that knowledge should be more than enough. And having said that, let's move on to the next stage. Now, this is a structure I'm not going to talk much, but all the concept which I've spoken about of the divine nature from beginning to the end is all present here. I'm not going to detail because you're going to see the beauty of it as you move on. But in this particular picture, you can see the beauty of 10 center chakras, then Ida, Pingala, Sishma 3. You can see the flight, you can see the thing, and other thing. I think this is enough for the time being. Thank you very much. I think lovely. Thank you.